think the answer is yes.
Okay, good.
So are you done now? Yes. We're done. No, beautiful. That's how good it's done for. No, here we go. So, who's
Food is an organic chemist, that's why.
yeah, okay, no, that's actually.
It's Joe's fault.
Don't have any advice. Okay, so thank you everyone for organizing this event and for the invitation to speak here. I have an unoriginal title. I think I did take this from Dan, How Do Enzymes Work? But I think it's a long-standing question. And so the idea of the trajectory and mixing the science is a great concept to present. I want to give you a little bit of a perspective of each and to build on some of the things Dan said at the beginning of the day. So to think about a trajectory for my path was I actually was born in the great state of South Dakota, in Sioux Falls. This is the Sioux River, and this is where it falls. I bring this up because, what's that?
The one place it falls?
Yes, this is it. Actually, there's a couple of mine. And I bring this up because I remember in one group meeting or a practice presentation for something, I said something foolish, and Dan's comment was, even though you're from South Dakota, you want people to think you're sophisticated. So you can be the judge of that. But after going to the local college here, I had a great fortune to go to the University of Chicago, where my graduate mentor, Joe Picciarelli, is actually in the audience here. And he was a fantastic mentor in every way possible to work. And nearing the end of my time in Joe's lab, he graciously allowed me to, and supported me to go to a meeting in Dundee, a ribozyme meeting. And that's where I first interacted with Dan. And he said, oh, you're nearing the end of your time. What are some of the labs and projects that you'd be interested in? I dropped a few names, no one in this room. And Dan said, we should get together later, and I'll tell you what's wrong with that. And so we did, and Dan was telling me some of the work they were starting or building on with using unnatural amino acids to look at enzyme function. And it was just this tremendous opportunity, and everything sounded outstanding. So when I sent the email expressing my interest, we had several email and phone calls and exchanges. And Mike Thorne said, at one point, Dan did say to me, you still have to come out to the lab to interview so we can see if everyone in the lab thinks you're a jerk. And so I visited, was given the opportunity to join the lab. I accepted immediately, and I was in Dan's lab actually from 2004 to 2012, and as a postdoc. Before the time came to move on, and there was an opportunity at the Cal State Long Beach down the coast here, where they're looking for a bioorganic chemist, whatever that means. And so I applied for the position. I was fortunate able to get that, and it's been a great experience in the past about 6 years, taking the knowledge and skills that I've learned from Dan at a place like Cal State Long Beach. It's a university about 37,000 students, about 1,000 parking spaces, it seems like. And it's a very diverse campus with a large number of underrepresented students, in particular in science. And on the teaching side, it's primarily been organic chemistry, and I've really been trying to use what I've learned from Dan to apply them in the classroom. It's not memorization, it's about thinking critically, thinking deeply about what you're doing. And also, in a research lab, working primarily with undergraduate students, this is a sampling of some of the students, really trying to inspire them to do excellent science, to do real science, and to think critically and deeply early on in their scientific development. I show this picture even though it's so unsanitary. Yeah. Not the best way to put their feet on the desk, but it was a nice capture of the group at the time. So, now briefly to try to present how this is being done, I'm just going to show an overview of 3 types of questions or projects that are going on in the lab. The first one is still an organic chemist at heart, for some reason, still find that interesting, of using organic chemistry to study enzyme function in 2 particular ways. One is that we have the great fortune of still working with Dan on a model oxygen whole system, and then second, long-standing interest in synthesizing unnatural amino acids. I won't go into the reasons as to why we're doing that today. So there have been, as Paul has mentioned too, a number of outstanding papers from Dan's lab about hydrogen bonding. I highlighted most recently Dan and Margot's perspective in biochemistry, which is such a succinct and outstanding read about energetics of hydrogen bonds. And we are now interested in this about basically going between enzymatic oxygen output studies and ketostable isomers and Paul and Shuro's early model work. And then Dan had asked, what do you think about continuing to work with us and developing this model oxygen whole system where we can have features of the enzymatic oxygen whole, but yet have the ability to systematically and incisively perturb these different properties. And I thought this was an outstanding opportunity also to train undergraduates and hopefully thinking about these energetics at this deep level. The first challenge, and this is almost where we're at now, is to synthesize this compound. Dan asked, can we make this? I said, of course, yes. looked at this paper, followed the procedures, disaster at first. And so after changing a number of plating catalysts and conditions, we had this Suzuki-mediated coupling and then we can readily deprotect and boron tribonate to make this compound out in hundreds of milligrams quite readily, so we're able to use NMR studies. I wanted to give you an overview of the different approaches. And so right now, this is what we'd like to see by NMR measurements. We're struggling right now in terms of trying to sort out the array of conditions, and so good for students training, bad for me as we're moving. It seems like a geological time pace on this. It's like rush hour on 405. But I think we're making steady progress, and using this as an approach to think that even something like troubleshooting, you have to think about it systematically, rigorously. What are the different models? I got an email from Dan just about 2 weeks ago. What are the models, right, for we were saying this maybe intermediate exchange? And so it's fantastic. So then, just skipping around a little bit, and there's a bit of a disconnect here, but I want to present something that I think it's in its infancy, but was an interesting find. In a way, this was an extra tube type experiment. So balancing, thinking critically, and developing models, but sometimes trying something out and see if you find something quite interesting. So as mentioned, still from where it started in Dan's lab, interested in making these unnatural amino acids in different and more efficient ways. This guy was interested in thinking about this enzyme butyrylcholinesterase and its inhibitors. So I don't want to overhype the role of this enzyme butyrylcholinesterase or butyrase, but there has been some interest in its potential association with Alzheimer's disease. By no means am I saying we're going to find a cure or something that can mitigate Alzheimer's disease, but to put it into context, for some time there's the polarogenetic hypothesis where these enzymes cholinesterases that are involved in neuronal signal transduction and chopping up the small molecules. So choline as it passes across the synapse. And it's not in patients with Alzheimer's disease. Perhaps one of these enzymes, butyrylcholinesterase, or butyrase for short, is actually higher in activity. There's little understanding of its function. It's highly related to acetylcholinesterase, which is involved in all sorts of processes. The structures of the two enzymes are essentially the same. And as I mentioned, in patients with Alzheimer's, HACE activity is essentially the same. It maybe decreases a little bit, whereas butyrase is increased. And one model is that increase in activity chops up too much of the small molecule neurotransmitter, and this may help contribute to dementia. So there's an idea of developing butyrase-specific inhibitors. And I tell this story in a way that makes us look much smarter than we actually are. But when you look at some of the current and past drugs that have been trying to target HACE in particular, Tacrin, which is now off the market, donepezil and this ribostigmine, both of these compounds contain aromatic moieties like almost anything, but also a carbamate, which is a functional group used in a lot of cholinesterase inhibitors. So one day, I was thinking about, what about our FMOC amino acids? These have this aromatic moiety and a carbamate. Do they actually inhibit? We have all these amino acids in the fridge. I had a student joining. I thought one of the first things to do is let's see if they inhibit and start working with proteins and enzymes. It's a very easy acid that we can do. So we took 19 of the 20 FMOC amino acids that's shown here by the one-letter code. We couldn't use cysteine because we have a sulfhydryl-based acid. It's colorimetric. And relative activity is activity without the inhibitor or with the inhibitor. So FMOC glycine does nothing. We chose a concentration under which all the potential inhibitors are soluble. And we found that a couple of compounds, leucine, tryptophan, and lysine, all inhibited. And I have to admit, I don't know why it's this at this time. I was thinking maybe this mimics a little bit of the substrate. This has these aromatic moieties, whereas leucine was actually the best inhibitor. So we're still sorting this out. This is qualitative. Quantitatively, these are reversible inhibitors. We've done experiments to test them. They have inhibition constants on the order of about 100 to 200 micromolar, so nowhere near useful, but hopefully a starting point to start thinking about structure. So I was really interested in what's important for binding in the enzyme, if they are indeed binding in the active site as a competitive inhibitor. One of the questions, as I mentioned earlier, was two enzymes, HACE and butyrase. Are these molecules specific? So we did the same experiment with acetylcholinesterase, and we found under the same conditions that there was no inhibition of HACE, whereas we were starting to inhibit butyrase. We've also tested this with chymotrypsin, another serine protease, elastase, and we chose these because of their similarities and they're also commercially available. It was quite easy to test these enzymes quickly. And now we're starting to tear this apart, thinking about what's really important for inhibition structurally. If we just have the amino acid, that's what's shown here by the dash, none of the compounds inhibited. If we trim back the aromatic moieties to keep the carbamate, these CBZ amino acids, to eliminate some of those, there's a little bit of inhibition, but it looks like the FMOC group in general is needed. And this has been hopefully a project now that can really start getting undergraduate students involved in thinking about working with a protein, thinking about careful quantitative analysis, learning about which end of the pipette is up. Experiments, reproducibility. So I've had a number of students now start to look at leucine was the best one, variations of leucine. They actually found this compound, neopentoglycine, is a better inhibitor by about five-fold, so maybe a little less. Now we're making peptides, making dipeptides and tripeptides. We're finding that these are becoming better inhibitors. What about changing this carboxylate? Maybe that negative charge could be unfavorable in an enzyme that binds a cationic substrate. So we're esтерifying to neutralize that. We're actually introducing a substrate feature like a polino group. And lastly, with stereochemistry, variation of the D and the L amino acids. And it actually turns out to be that the D amino acids are better inhibitors. All the expensive ones are the best inhibitors. And I have to say that this has been a great project working with undergraduate students because they have their own project to take ownership and to work on. We're not on an island because we're doing similar things. And we can really get to compare values and thinking about experiments with each other. So then in the last 30 to 40 minutes here, I wanted to just quickly mention one other work that has been going on and thinking about near active site features. So we all know that an enzyme contains many more residues than those residues that are just directly involved in making and breaking bonds. And even taking an enzyme like triose isomerase, where there are these 3 active site residues that have been studied extensively, when you look at a comparison of all of the known sequences of TIM, what you end up finding is that there's this other glutamate residue here highlighted in the blue, glutamate 97, that's found in all of these sequences. So ask the simple question, what's the catalytic role of this residue? I initially got interested in this because it's next to the very important lysine. The idea was that I was going to be positioning, it may be positioning that lysine for catalysis. Then we did some looking in the literature and found that there's a paper that came out in 2011 where this glutamate 97 was mutated to aspartate, about a 10-fold effect. Sorry, Justin, I'm showing KCAT here. I had a choice between KCAT and glutamine, and the activity was down about 3 orders of magnitude. These folks rewrote the mechanism of TIM, including saying that this residue is now directly involved in catalysis and proton transfer. John Richards and others disagree with that, and there's been a debate back and forth in the literature. But then looking further back from the chicken enzyme, the same glutamate mutation was made, and there was no rate effect. This is just buried in the table from the moles. So when you look at the 2 structures, of course, these are both tin barrels. The hallmark of the name is the one that has the larger effect is from this plasmodium, and then the chicken enzyme has the little effect. When you look at the structures, though, even though they're both tin barrels, in white I'm showing the plasmodium, the one that has the larger effect. In the chicken enzyme, where there's no effect of this mutation of the glutamate, you see there are actually a few differences in the surrounding residues. Same overall fold, of course, can be active in different ways. In particular, this phenylalanine is a serine, this serine is an alanine, and this is asparaginase is glycine. So we started looking at some of the different organisms that have been studied before, and the chicken, this is a trypanosome TDD for short, that's been studied quite a bit. Yeast, and then the human TIM enzyme has been used in some experiments. And we actually had the plasmids for a few of these in the fridge, and so the idea was maybe these differences in these active site features may be leading to these different mutational effects. So I was able to convince Tim Chang, who I assume was always named after the enzyme, Tim, and it made for confusion in the freezer. So anyways, each for the yeast and for the human, and then the chicken enzyme, I've shown this slide once to recruit undergraduates and graduate students. I had maybe you. Yeah, so I was going to try it. If anyone's in Long Beach, let me know. So I took it out, but one of the first challenges, and this was actually a monumental effort, was going from almost an empty room to making and expressing and purifying tin and getting reproducible kinetics. One of the things in the literature is this is one that's been highly studied, but the KCAT values vary by almost 3 to 5-fold in the literature. It's slowing down over time from 1,000 to below 300 per second. And John Richard actually very nicely mentions in the paper, we don't understand why that's changing. So
it goes against global warming.
Yeah. But I was able to have Tim over months make the enzyme multiple times, independent preps, and we're getting values similar to the Richard point, more recent values. And we did some engineering in the yeast and the human, and a little molecular biology to get tags on them to purify them. So the bottom line is when we make these mutations, we also make an alanine mutation in each of these different organisms. The mutations actually decrease relatively to modest effect. Almost no change. Again, I'm showing KCAT, but these are mostly KCAT effects. There's a slight increase in K on the glutamate mutation. And there's also almost a tenfold mutation. And on top of that, the aspartate and the glutamine mutation are very similar. Not that tenfold and 1,000-fold that's been seen in the plasmodium. So we then turn back the structure, and this is a bit of a hand-waving argument at this point, and look to see what's the difference. So in the plasmodium, this serine, is a phenylalanine, but in all three of our enzymes, and I'm only showing TDB, the three enzymes we tested are all the same. This, as I mentioned, this phenylalanine is a serine. Fortunately, the folks who did the prior mutagenesis study in the plasmodium solved crystal structures of their 2 mutants, but they didn't really comment on this. And they actually see that this phenylalanine is shifted by almost 3 angstroms. Mutation of this residue actually results in a deleterious effect. So our current model is that the mutation of the glutamate to an aspartate, notice how it almost sits in the same spot and make the same hydrogen bonding interaction, leads to that 10 to 100-fold rate effect due to now mispositioning of this adjacent phenylalanine. The glutamine mutation in the plasmodium, and I should also reference that we have a little serine there so that we're not seeing that deleterious effect. The glutamate to glutamine mutation shows that the residue is situated in about the same position. Again, perhaps make these hydrogen bonds. Now the phenylalanine is attacked, but the adiposite lysine goes from its position in the wild type to that E97Q mutant to shift by again almost 3 angstroms, and maybe making hydrogen bonds with an asparagine and a serine, the side chains, where our enzymes, that's a glycine and an alanine. So that you've now actually maybe introduced a favorable misposition interaction here. And if our enzymes, they don't have those positions. Now, what we really want to do is carry out the same experiments in the plasmodium and do some double mutant cycle analysis to see if this holds up in the activity assays. And it would be really great to get crystal structures of our particular enzymes. So the bottom line here is even though the overall structure is about the same, different surrounding residues can give different mutational results. And so we're thinking about this in both just a fundamental understanding of how enzymes work, but also in terms of one wants to design an enzyme, thinking about incorporating interactions that are either favorable or trying to avoid interactions that are unfavorable. So this leads to one last question, I'm just about done, Marcello, is that why is this glutamate residue found in all of these sequences of 10? And it is actually near the dimer interface. These are the 2 monomers. It sits near this where this loop is interacting with the glutamate. We blew a lot of our money on the top line gel filtration column and that. And we actually think that, or maybe this is affecting dimer stability, and we're actually seeing some that looks like disruption of the dimer in those mutants. And that's work that's ongoing right now. So following from this, non-active site or second shell residues are important. There's many different ways to get to the same structure. This is taking language from Dan, I think, whereas it's more difficult sometimes to assay these because information about chemical mechanism is not as apparent. You can think of I need a general acid, I need a general base, based on the mechanism, but the surrounding residues can be much more difficult to evaluate the results without experimental data. So, to summarize, hopefully I've given you a flavor of a few of these types of projects that are going on down at the beach. And last, I want to say once again thank you to Dan for all of his infinite wisdom, advice, letting an organic chemist kind of slide into the lab. Overall, sometimes when we talk about protecting groups and in Dan, it's exactly what I would do, is use that slide. And I have to admit, with what Paul had said, I actually used that line on one of the students in my lab within the past month about the disservice to everyone who has come before them. It was their first week in the lab, too, wasn't it? So last, then, it's a pretty vibrant environment. I've had a great chance to work with a variety of students. And one of the things that's nice is that hopefully I can convince you, taking science and the approach that Dan has taken at a primarily undergraduate institution, to provide opportunities for students. These are programs from the NIH and Department of Education to help increase the number of underrepresented students in science, and taking Dan's approach and applying it down there. So thank you.
Neat. So if you took those 2 enzymes that I'm going to blank on the name, so falciparum and PDB, I guess. So, and if you took the one enzyme and made the surrounding residues the same, maybe you did this and I missed it, then made the mutation of the 97, do you see the same effect?
No, we haven't done that. And so that's a great suggestion.
One question.
Is your 5 to 10-fold effect of KCAT, could that be related to dimerization and people using different buffers, different enzyme concentrations?
There are a lot of differences in the literature, but when you look at a variety of papers, even from the same labs, there's been some variation. I think the prep has been different too initially, so it's likely the earlier enzymes, no, not blaming them. When you look at the protocol, there could have been some issues with actually quantifying the enzyme. We've even spent a lot of time working to make sure that the enzyme's not sticking in the tubes and tips and things like that. And done some of those really exciting but very necessary controls.
Who's got definitely? Go ahead. So Ed, first on, where's Pat? So what was the variation in the literature for alpha phosphatase?
They all were perfectly agreed. Perfect.
It was 4 orders of magnitude, is that right?
Only 3 orders.
3 orders of magnitude.
So that makes our effect really small.
Yeah, exactly.
Threefold.
Well, so any idea why the phenylalanine movement would cause a difference?
So one possibility is actually it's adjacent to the negative charge near the potential anion-aromatic interaction. So it is actually situated.
And then, yeah, so isn't that glutamate that you mentioned, is that really glutamic acid?
So there's a debate about the protonation state.
Sorry, that was a setup for Margot. Margot, you want to know wherever she is? Is that a glutamic acid?
Are you talking about the dimer interface?
Yeah.
Yeah, that's a different position, but yeah.
But actually, it's been shown as both in the literature, actually. And so I kept on saying glutamate here, but I've seen both. And most of the time, where it's situated, though, when you have hydrogen-bound donors and acceptors, a lot of these things are like the serine as well. And that's actually where we have phenylalanine, where there's an asparagine. You can have either the side chain flip, or you have a donor acceptor, so it's a little hard to delineate the network. I've been trying to design as to what the possible models for the networks.
So Margot knows how to figure this out from metagenomic data, so she can tell you about it.
That's probably better than me and Pynol.
I was just going to ask, because I might have missed it. Do you have any kind of kinetic data or anything to suggest that it's actually binding to the active side? I might have missed it.
For the cholinesterase inhibitors, all we can say, we've been looking and seeing if the inhibitors are competitive or not, using the calcium, I think, right? And these look like they're mixed, actually. We have some other ones that show the behavior of a simple competitive inhibitor, but these do not show quite that.
So these are great projects for undergrads. And, I had many thoughts as you were going through this. The fact that you're working with a malaria enzyme makes my heart warm, because it's not because of any fantasies about curing malaria, but undergrads becoming interested in something, in developing networks and things like that. I think it's terrific. So if you ever want a list of people who make good enzymes and have decent assays or something, you have all these compounds. This could be a pipeline that could keep going for a while.
Yeah,
And there are friends who work on TB and other important things, so keep that in mind if you want to go down that road.
That's fantastic. I have to admit, always wanting to focus on the questions in the science, convincing students to work on an enzyme associated, say, with Alzheimer's, that has been associated with Alzheimer's disease, is oftentimes an easier sell than the energetics of hydrogen bonds. The eyes glaze over. We haven't taken organic too.
Did they read Schro's paper? Don't they all read
So have to read the paper and figure out which end of the pipette is top.
Okay. There are no more questions. We have like 2 minutes for coffee break. I think 10 minutes, and then for our generation, the third generation, we can take a picture. But we want to take a picture.